Many signal transduction pathways ultimately change transcription levels of genes in the nucleus. Some do this by sending a kinase through a nuclear pore into the nucleus, where it can phosphorylate a transcription factor which can then bind DNA. So, for example, in the MAP kinase pathway, the MAP kinase ERK2 translocates to the nucleus, where it phosphorylates the transcription factors c-MYC and ELK1, which promote cell division. Another MAP kinase pathway results in the MAP-like kinase JNK enzyme moving to the nucleus, where it can phosphorylate c-Jun. Many hormones activate the second messenger cyclic AMP, which in turn activates the kinase enzyme pKa, which can move to the nucleus, where it can phosphorylate the transcription factor CRIP. In some pathways, a cytoplasmic kinase enzyme phosphorylates a target in the cytoplasm, and it is this target which then passes through the nuclear pore into the nucleus, where it acts as a transcription factor binding DNA. So, for example, cytokine receptors can activate JAK kinase, which phosphorylates the effector STAT in the cytoplasm, which can then pass into the nucleus and bind to DNA. The TGF-beta signaling pathway phosphorylates a cytoplasmic target SMAD, which moves to the nucleus, where it can bind DNA as a transcription factor. Another way that transcription factors can be activated is if the enzyme in the pathway, such as the cyclin-dependent kinase depicted here, phosphorylates a repressor protein. In this case, the tumor suppressor retinoblastoma, the RB protein, has bound to the transcription factor E2F. And while retinoblastoma is bound to E2F, it cannot act as a transcription factor promoting the transcription of the genes which will promote cell growth. Once RB is phosphorylated, it disassociates from E2F, which is now free to act as a transcription factor binding DNA and promoting cell growth.